Welcome everyone to today's endocrinology grand rounds. Before I introduce Dr. Kling, I just wanted to mention a couple of things. Um, Next week, we will be having our a visiting speaker from University of Illinois in Chicago, and he'll be giving two lectures. One will be the endocrine grand rounds, and the other is the medical grand rounds, which is the Tawari lecture, which is in honor of Dr. Beverly Todd Tawari, who was chairman of medicine and chief of endocrinology here for a long time, from 1956 to 72 or so, or longer. So we invite everyone to attend both of those lectures. It'll be on environmental impacts on metabolic health. I don't know if we can still talk about it or not, but we hope so, you know. Um, so the lecture will be not in this hall, but it'll be in the auditorium right across there. So uh, for today, we have Dr. Kling, who will be talking about the role of M6A in RNA transcripts and endocrine therapy resistance in breast cancer cells. Dr. Kling really doesn't need any introduction to all of you, but I just wanted to mention certain things. One, I mean, she's professor of biochemistry and pharmacology at the University of Louisville. She has been at the University of Louisville since '96, um, and moving up from being assistant professor to being a full professor. She has been a great asset for the endocrinology division because she is in charge of our molecular and cellular endocrinology series, which she you know, gets excellent speakers and excellent topics for us. She has been a great teacher because she has mentored people from high school through postdoctoral fellows. There are numerous people who have trained under her and have benefited from her knowledge and expertise and counsel. And she has also spoken here several times on different topics related to endocrinology. So we always appreciate the work she puts in to get speakers, and also, we have had collaborations with her. She's also one of the PIs on the T35 grant related to endocrinology training. So we are very happy that she has been continuing to get that and support endocrine training for medical students. And so, Dr. Kling.
Thank you very much for that kind introduction, Dr. Mokshoon. Okay, so let's get started here. This work was supported by an NIH grant, as you can see, by a Department of Defense Breast Cancer Research Program grant, and also by a grant from the Jewish Heritage Fund for Excellence. And I have nothing to disclose. I have 4 learning objectives that were in the seminar notice, and I think you'll be able to complete these learning objectives if you listen to what I say today. So first of all, what's epitranscriptomics? What epitranscriptomics means is above the transcriptome. So these are chemical modifications that occur on all transcribed RNAs. The most modifications are seen in tRNAs and then ribosomal RNAs, followed by mRNAs. And these are some of the modifications. They occur on all 4 nucleosides, as you can see. And the most common one in transcribed mRNA is N6-methyladenosine. So that's the one I will focus on in today's topic. But if we look at an mRNA transcript, we can see M6A both within the coding region as well as in the 5' untranslated region in the form of M6AM. Actually, there's 2 methyl groups there instead of 1, as we see in the coding region, but it's also in the 3' UTR as well. So really this diagram is not completely accurate. But what is interesting is that just recently in 2024, a paper was published showing that M7G actually occurs within the coding region and 3' UTR, as well as at the end M and terminus of the transcript, the 5' end of the transcript, which is important for cap addition onto the transcribed RNA. But still, M6A, which is the most common modification in mRNA, is still a rare modification. Only about 0.1 to 0.4% of A's are actually modified, and that modification occurs within a specific nucleotide region that we'll see on the next slide, I think. But we have an average of 3 to 5 M6A sites per transcript. And this has become a very interesting topic for research. Starting at about 2015, you can see the number of publications per year, including M6A, has taken off. And that's about when I got interested in M6A as another level of regulation of the transcript to become a protein and therefore mediate its biological activity. Now, fortunately for me and my collaborators, there are fewer publications about M6A in breast cancer. So we're in a good position, and Mary Sumlett, who's a graduate student in Dr. Brian Clem's lab, knows all about that as well. So this diagram features the mechanism by which N6A is methylated. And so what happens is that there is a complex of proteins called the Reiter complex, which includes metal 3, which is the catalytic enzyme, the methyltransferase that takes the methyl group from S-adenosylmethionine, a metabolite, so that gets into nutrition and well-being, and adds that to that 6th position on adenosine to form M6A. Now, this is a dynamic modification in that it is attached by M-metal 3 complex, but it can be removed by demethylases, namely ALK-BH5 and FTO. Now, what M6A does is it allows the binding of RNA binding proteins, and there's 18 different RNA binding proteins at this point that are known to bind that M6A mark. And their binding then conveys different properties to that M6A decorated transcript. And so the writers can either be involved in splicing of the mRNA to different forms, or in terms of processing of a primary microRNA transcript into its precursor microRNA transcript. M6A readers can be involved in the export of transcripts from the nucleus into the cytoplasm for translation. They can either stabilize or destabilize transcripts and therefore regulate protein availability in the cell. So why do we care about epitranscriptomics? I'm interested in endocrine therapy resistance because it's a real problem for breast cancer patients, most of whom have estrogen receptor positive tumors. 33 to 44% of those patients will develop endocrine therapy resistant metastatic disease with progression. So my question is, what's the role of epitranscriptomics in acquired endocrine resistant breast cancer? So these are patients who have developed resistance to tamoxifen, to fulvestrant, which is used in metastatic settings, and to aromatase inhibitors. So to do this, we use a breast cancer cell line model. And I realize this is kind of busy, but if we go from left to right, what we see are tamoxifen sensitive cells, namely MCF7, T47D, and ZR75-1. And these have been selected by pressure either in tamoxifen or raloxifene to develop resistance to endocrine therapies. And so these are the T47D 4-OHT, ZR75-1 4-OHT, respectively resistant compared to their parental cell line. And then MCF7, LCC9 and LY2 were derived by Robert Clark at Georgetown University, who gave us these cell lines. These T47D 4-OHT and ZR75-1 4-OHT we've developed in our lab right upstairs. So the first question we asked is there a different expression of the readers, writers, and erasers of the M6A mark in endocrine-resistant versus endocrine-sensitive breast cancer cell? And we looked first at the transcript level, and the answer was yes. We see that some readers and writers are increased in expression in the endocrine therapy resistant LCC9 cells compared to the endocrine therapy sensitive MCF7 cells, and some are down regulated. And interestingly, we saw that IGF-BP2, which is a reader involved in mRNA stabilization, was increased in LCC9 endocrine therapy resistant cells, whereas YTHDF1, another reader that is involved in translation initiation or mRNA decay, depending on the particular transcript and cellular abundance of the reader itself, it was downregulated in LCC9. So we confirmed that by our real-time quantitative real-time PCR, and then by Western blot analysis. And Kelly Peel has done many of the experiments. She is a master technician and lab manager in my lab, and I'm very glad to have worked with Kelly for a number of years now. So what's the importance of IGF-BP2? Well, it promotes breast cancer progression, and its transcript abundance was also higher in another MCF7 tamoxifen resistant cell line compared to the parental cells or the MCF10A cell line, which is considered a model for mammary epithelial cells. So what are we going to do now that we know that IGF-BP2 is upregulated in our endocrine therapy resistant LCC9 cells? Well, we want to find out what transcripts it's regulating. What are those M6A transcripts that IGF-BP2 binds in LCC9 that it's not going to bind in MCF7? So we have done an antibody, what's called a RIP, RNA immunoprecipitation experiment. So we have an antibody to IGF-BP2 that binds the IGF-BP2. The IGF-BP2 is binding the mRNAs, which have the M6A mark, and we can then use that negative control IgG, and we can treat the cells with an inhibitor of metal 3 called STM2457, which we'll come on to later, so we can differentiate between non-specific and specific M6A marks. And then we can run that RNA in Illumina RNA-seq. Now, one issue with Illumina RNA-seq is a short read sequence, but you get a lot of data, is that you have to convert the RNA to cDNA, and that can introduce errors in what your readout of the RNA would be in this situation. But we've done it all, and we sent our samples to the Sequencing Technology Core, which is now over on the 6th floor in HSCA in October of last year. We have no data yet. So I want to say that if you're interested in these RNA-seq types of experiments, you have to have a great deal of patience and think ahead of time, because it's going to take you a while to get your data. But our goal is to identify what are the transcripts bound by IGF-BP2 in LCC9 endocrine therapy resistant breast cancer cells, because these could be novel targets for blocking endocrine therapy resistance in breast cancer. So now back to our main question. What's the role of M6A in acquired endocrine resistant breast cancer? Again, now we're going to simplify our breast cancer cell line model, and I'm going to focus on MCF7 as endocrine therapy sensitive versus LCC9 as endocrine therapy resistant. Those other cell lines we'll use in confirmatory experiments that you will see later on. Okay, so what we did was to treat MCF7 or LCC9 cells with either DMSO, which is dimethyl sulfoxide. That's a vehicle in which 4-hydroxytamoxifen, the active metabolite of tamoxifen in the 100 nanomolar concentration, is a concentration that is found in circulation and in tumors of patients who take oral tamoxifen citrate, premenopausal breast cancer patients with ER-positive disease. And then we also treated cells with 1.5 micromolar STN2457, an inhibitor of metal 3, that methyl transferase. So we should see a decrease in M6A in those samples. We did 5 biological replicates per cell group, and we performed direct RNA sequencing. Now, the beauty of the direct RNA sequencing is, as its name implies, direct RNA sequencing. You don't have to do the step of converting the RNA to cDNA for sequencing like you do with Illumina. And so what happens is you put the RNA into a device called a MinION nanopore. And that has an array of scaffolds and a sensor chip, and the RNA is read through the pores, and you get an electrical signal, which can then be converted to the reads of the RNA. And from the electrical signal, you can specifically appoint a site at which the M6A methylation occurs. And that is also unique because there's other techniques called MIRIP or M6A RNA immunoprecipitation that are used to identify where M6A is located in transcripts. But unfortunately, that's an immunoprecipitation. And again, once you have your immunoprecipitated RNA, then you have to transcribe to cDNA or reverse transcribe to cDNA and then sequence in Illumina. So again, you have steps at which errors are introduced. So this is just a summary of all our samples. And one of the things I want to point out is we have used two different flow cell kits, those chips with the pores for direct RNA sequencing. We started with the RNA 002, and now the current one is RNA 004, and there wasn't an RNA 003. Must have failed it somehow. But so we do have two different flow cells that we used. And the good thing is that this RNA 004, which was new early last year, is that it has much higher read density. Actually, I should say the middle of last year. Okay, so what happens now that we have the signal from the nanopore? You get FASTQ and FAST5 sequences that go through a number of steps and ultimately through a detection algorithm called M6ANet that was selected by Dr. Rushka as the best program to analyze the FASTQ, the direct RNA sequencing, and identify specifically where the M6A sites are in the transcript. And what Dr. Rushka and Dr. Belinda Petrie, who has worked very diligently on this, the output is probability of modification and the modification ratio. Now, one thing about this M6ANet that's very interesting is it takes 5 days to run per sample. So we have a lot of samples in 5 days per sample. That's the computational time it takes to run this program, this algorithm. If anything goes wrong during that 5 days it's running, say there's a little power glitch, a little surge, a little power outage, it doesn't stop where it was reading. It has to go, you have to restart the whole thing. So this has taken a lot of computational time and effort to annotate what we have. So our two questions are what are the M6A sites in transcripts in the endocrine-sensitive MCF7 versus the endocrine-resistant LCC9 cells? And which of these M6A sites is regulated by metal 3? Because that should be a direct regulation mediated by the M6A deposited by metal 3 that's going to be inhibited by the compound STN2457, a specific inhibitor of metal 3. So what do we see? So now Belinda has done all this computational annotation, and one concern is, of course, false positives. And so you can set different p-values and find out how many M6A sites are significant. So what she did was set different threshold values, and you see that in this volcano plot here. So this is looking at LCC9, endocrine resistant versus the endocrine sensitive cell line. And depending on your cutoff, so if you use a p-value of 0.05, you get 7,000, 0.01, 1788. And what we have used, or she selected for our annotations and subsequent follow-up experiments, is a p-value of less than 0.001, and that gives us 362 M6A sites. So where are these M6A sites? Well, most are in the 3' UTR. Some are in the coding sequence, and some seems to be in the 5' UTR. Now, this M6A net is looking at the sequence at which it detects that sort of little, I'll say, electrical blip that might mean M6A. And it will say, that's probably an M6A with differential p-values if it's located within what's called the DRACH sequence. I failed to mention that. So when the metal 3 complex is looking for an A to methylate, it has to be within the nucleotide sequence called the DRACH sequence. And it's an invariant AC, so that A is the target of the metal 3 to add the methyl group. And then various nucleotides specifically flanking that AC, but invariant AC. And so it's not surprising that we see A in the DRACH sequence by our analysis, because that M6A net that we're using as the algorithm to read the electrical output from the ONT nanopore device is looking for that DRACH sequence. But nonetheless, all good. This is proving that we're being honest and successful in our demonstration of specific M6A methylation within transcripts. So then the question is how many M6A modifications do we see per gene? Remember, it said like 3 to 5. Well, most of our genes have between 1 and 5, so that's in agreement with the published literature. And the published literature is from M6A RNA immunoprecipitation, that Illumina cDNA, not the direct RNA sequencing that we're doing. So, and there's really not a lot of difference. Perhaps there's more transcripts in MCF7 that have more M6A modifications, since we see a higher orange bar here, 6 to 10. So then the next question is, what are the differentially expressed genes in MCF7 versus LCC9? And what Belinda shows in this heat map and in the volcano plot is there are 11,370 differentially expressed genes, about equal numbers up and down regulated between MCF7 and LCC9 cells. Metacore pathway analysis can look at those genes both up and down regulated and find out what pathways are associated with those changes in gene expression between LCC9 and MCF7 cells. And what we can see is Wnt beta-catenin signaling and Notch signaling. And these are known pathways that have been implicated in breast cancer progression and tamoxifen resistance in vivo. So our cell model is actually a good and strong one. So then we wanted to know, what are the differentially expressed genes with M6A differences in MCF7 versus LCC9 cells? Well, if we flip that around, LCC9 versus MCF7 cells. So this is an integrative analysis that Belinda did to look at the RNA-seq data and the direct RNA-seq data. So what genes are associated with changes in M6A. And what she found is that genes that are changed with M6A modifications in LCC9 versus MCF7 versus the differentially expressed genes, there's only an overlap of 74. And she found genes that showed increase in M6A with increased transcript abundance, and every combination of imaginable. So decreased M6A, decreased gene expression, increased, decreased, decreased, increased. So what does this mean? Well, again, Metacore pathway analysis, which is an online program to which UofL has a site license, so you can run your data through this, shows regulation of skeletal rearrangement cell junctions, actin fibers. These are processes that are involved in breast cancer progression. So this fits again with our model that endocrine resistance is breast cancer progression. So what are then the differential genes with M6 modification that are associated with endocrine resistant breast cancer? So what Belinda and I did, and really Belinda was the lead. I just have my end note from many years with about 46,000 references in it, and looking for the genes that are changed in endocrine resistant breast tumors. And so then looking at those differentially expressed genes, so those, oops, if I go back. So looking at these 74 genes and seeing which of these 74 genes are known to play a role in endocrine therapy resistance and progression and metastases in breast cancer. And so we have a list of genes that you can see here. And we have picked out some that we want to look at. We are going to look at actin, beta-actin, ActB, EEF1A2, FLNA, and PDIA6. So the ones with the green arrows are downregulated in terms of M6A in LCC9 versus MCF7, and downregulated at the transcript level, whereas PDA6 is up and up. So let's see what we see at protein. So the level. So EEF2A is an eukaryotic translation elongation factor, and it's known to be an M6A target. So this is like almost a positive control in our experiments. We have pulled out this transcript that other people have reported to be M6A decorated and modified as well. So Kelly ran Western blots of these proteins, and what we can see is that in fact we do see an increase in PDI1A6, a decrease in beta-actin and LCC9, a decrease in the EEF1A2, and a decrease in FLNA in LCC9. So these results agree in direction with that which we had seen in the differential gene expression. So the question then becomes, well, okay, do these reflect changes in human breast tumors? And the answer is yes. There's a website called TMN Plot that has human breast gene chip data. So you can put your gene in there and compare its expression levels in normal, in breast tumors, and in metastatic breast cancer. And what we can see is we see an increase in PDIA6, so it's increased in breast metastases, actin, beta-actin, and EIF1 are increased in the tumors, in primary breast tumors, and in metastases compared to normal breast. And FLNA is increased in the breast tumors, but it's not further increased in the metastatic breast tumors. We think of endocrine resistance as promoting metastasis. So we want to know if our data are in agreement with what's out there and known in human breast tumors. And then the next question is, well, okay, so you compared MCF7 and its sort of daughter cell line that's endocrine resistant, LCC9. What about other breast cancer cells that have developed that are models of endocrine therapy resistance? Well, some of our data agrees and some of it does not agree, but in general, we found agreement with other endocrine therapy resistant cell lines, including LY2, the T47D 40HT, and the ZR75 40HT cell line. So now we know where M6A sites are in transcripts, and we know that they're associated with differentially expressed genes. How can we test if a differential M6A modification regulates a particular transcript or the abundance of that protein? I mentioned the STM2457. There's the molecular compound structure of STM2457, this selective inhibitor of metal 3. So we should see a decrease in M6A with using this compound. So first we had to select a dose of STM2457 that wasn't going to kill our breast cancer cell lines. So Kelly did MTT assays to look at the viability of either MCF7 or LCC9 cells in response to increasing concentrations of STM2457. And we determined using graph-fed PRISM the IC50 value for each cell line. You can see it's about 60 and 91 for each cell line respectively. So we selected to use 1 micromolar STM2457 for 48 hours unless otherwise indicated for follow-up experiments here. And then we asked, okay, does STM2457 really decrease M6A in RNA? And the answer is yes. We can see that here. We see a decrease in M6A. Now we're using this concentration. It still is going to leave M6A on, but it's not inhibiting the viability of the cells. So how does STM2457 affect transcript abundance or differential gene expression? So we have our heat maps, and then we have a Venn diagram to show the differential gene expression in MCF7 cells treated with STM2457 or the vehicle control versus LCC9 with STM2457 or the vehicle control. And we see those that are specifically regulated by STM2457 and therefore M6A within each cell line and commonly regulated between the two cell lines. And we see transcript abundance both increased and decreased. And again, this has to do with the fact that M6A can either stabilize or destabilize a transcript. It can increase translation or decrease translation. It depends upon the specific transcript. It depends upon the reader. It depends upon the cell line. So what pathways then are implicated in M6A regulation? At the top, you see the pathways from MCF7, and at the bottom, you see LCC9. Some are the same pathways and some are different. And also in LCC9, interestingly enough, includes estrogen receptor alpha and beta in membrane signaling. So these are genes that are enriched in similar pathways that are altered in endocrine therapy resistant cells. So next, what Belinda did was to do an overlap analysis of the STM2457 differentially regulated genes. So remember, we had that 74 differentially expressed genes with M6A in LCC9 versus MCF7 cells. So then she split that into cells that are upregulated by STM2457 in MCF7 or downregulated by STM2457 in MCF7 cells versus differentially expressed genes in LCC9 with STM2457 upregulated or downregulated, and then integrated that. So now we have those transcripts that should be directly downregulated by STM2457 and MCF7 cells and versus directly downregulated by STM2457 in LCC9 cells. And we have one gene in common, that EEF1A2, that we have seen before. So we looked at how STM2457 would affect those transcripts in a separate RT-qPCR experiment. And what we can see is we see downregulation of actinomycin D, of keratin 18, of NRAS, but not significant of EEF2, even though we detected it in the differential gene expression in the Illumina data. And then for on the LCC9 side, so this is from the right side and from the left side, green, peachy to peachy, cited D, TPT1, XBP1, it's a transcription factor, and EEF2. Again, this is not statistically significant for EEF2. So then we pulled back and said, which transcripts are STM2457 regulated? And one was of interest to us because we can see that it's STM2457 regulated, and that is amigo-2, increased with STM2457 in both MCF7 and LCC9 cells. So that means that lower amigo, M6A, gives us more amigo 2 because we've inhibited the methyltransferase M-metal-3 with STM2457. We see more expression. So we're getting more amigo with less M6A. So our hypothesis is that when there's M6A there, it's decreasing transcript stability. So amigo 2. Now, this really started when Bailey Valdez was a summer student in the lab as an EMS2 student. She's now in her clinical third year. With looking at amigo 2, which is adhesion molecule Ig with like domain. And it functions in cell adhesion via plasma membrane cell adhesion molecules. It's a negative regulator of programmed cell death. And it's predicted to be located in both nucleus and in plasma membrane. If you do string, this shows the other proteins with which it interacts. And this also shows other proteins with which it interacts. Interestingly, it's upregulated and oncogenic in a variety of cancers. And this was a review that was published in Oncology Letters last year. But its function in breast cancer is unknown. So we're still at the vanguard of understanding amigo-2 in breast cancer. And the 5' UTR of pre-amigo-2 RNA was M6A regulated by YTHDC1, one of those M6A reader proteins binding that increased splicing in esophageal squamous cell carcinoma cells. And that was published in 2024. So other people are starting to look at amigo-2 and its M6A methylation in cancers as well. All right, well, what about breast cancer? You run KM plotter on any gene and find out if your gene is associated with overall survival beneficially or not. So if you look at, you can plug in various parameters in KM plotter. So I plugged in ER positive, HER2 negative breast cancer. And these are data on the left from Affymetrix array. So these are kind of older data. And what it shows is that low amigo-2 is protective and high is slightly lower overall survival, but the p-value was not that great. That's 0.05. So, okay. But they also have updated data from RNA-seq in KM plotter. And again, I put in the same parameters, ER positive, HER2 negative. And now what we see is that there's a slight advantage for higher expression and a lower p-value. So this points to that maybe amigo-2 is a tumor suppressor. And in fact, there was a paper published in 2021 that found that amigo-2 was one of 4 resistance-related genes associated with disease-free survival, and that amigo-2 was downregulated in advanced staged breast cancer. So this would sort of agree with our LCC9 model that we're looking at. And what about the TMN plot? Yes, right, there it goes. It's down in tumors and down in metastatic disease versus normal. So it's looking to us like amigo-2 is a tumor suppressor in breast cancer. All right, and back to Bailey's days in the lab. She and Kelly ran Western blots, and of course they did RT-qPCR and showed that amigo-2 is in fact lower in LCC9 compared to MCF7 cells. So this recapitulates what we saw in the Illumina differentially expressed genes. And it's lower at the protein level in LCC9 versus MCF7 cells as well. So endocrine resistant cells have lower amigo-2, lower tumor suppressor amigo-2. So then Belinda looked very carefully at where the M6As are located in amigo-2. And you can see that there are 3 different transcripts of amigo-2, and these are the 3 transcripts down here as well. So these are splice variants. And we have M6A sites located within the coding region of amigo-2. And we picked this position 670 for our first analysis of what specific role might that M6A at position 670 within the coding region have? So first of all, we wanted to validate that M6A was really at that position. The data from the nanodrop fed through M6A net showed, I go back, M6A at position 670. How do we know it's really there? Well, one way you can demonstrate that it's there is to do what's called MERIP. And again, this is M6A RNA immunoprecipitation now, real-time quantitative PCR. So we have an antibody to M6A, so it's going to bind to M6A, and you make primers flanking or overlapping that M6A sites, and you pull down the RNA versus the cells treated with STM2457, which should remove that M6A mark or not have it deposited. And what do we see? So if we look at fold enrichment in MCF7 versus LCC9, first of all, as good biochemists and molecular biologists and clinicians, you always look at the y-axis. So we have a lot more amigo-2 transcript in MCF7 versus LCC9, which is what we saw in the differential gene expression, which is what we saw in the protein, which is what we saw in real-time quantitative PCR. And lo and behold, STM2457 reduces the interaction of the antibody with the M6A mark on this region in position 670 in amigo-2. So that demonstrates that that site is methylated. Yay, at least in MCF7. But what about LCC9? We saw it there as well. Is it important? Well, again, now we don't have a lot of fold enrichment because there's less transcript and we've got a lot of scatter. So is it below our sensitivity to be able to detect it there? Maybe, right? So our follow-up plan is to investigate all the other M6A sites. And if we go back, well, we've got a lot of sites to look at in amigo-2. Maybe there's some sites that are more important in LCC9 than they are in MCF7. And we just lucked out by picking the 670 and found that that one is important in MCF7 cells. So when Bailey comes back to the lab in November and does her DIR work, she'll have lots of fun experiments to look at. Okay, how else can we determine if M6A and amigo-2 mRNA transcripts has a role in transcript stability, right? Well, I read this paper, and there's the paper, and this BNIP is BCL2 interacting protein. It's a mitochondrial pro-apoptotic factor, but it doesn't matter what the gene is. What they did was they developed an assay. This group developed an assay using pMIR-GLOW. So pMIR-GLOW is a promega reporter that has two luciferases. It has firefly luciferase with a multi-cloning region downstream from the firefly luciferase and ranilla luciferase. Each of these are driven by strong promoters, but the influence of you can clone, like you could clone a microRNA response element in here, but they cloned the BNP 3' UTR into it. And the BNP 3' UTR had 3 Dratch motifs, and then they mutated one, the first one, the second one, or the third one. And they did a transient transfection into a breast cancer cell line, a triple negative breast cancer cell line, with either control or they knocked down an eraser, FTO, and they did 2 different constructs of the eraser. But the big thing to look at is that if you knock down FTO, you're removing the eraser, so you're increasing M6A. By increasing M6A, they saw an increase in luciferase activity. So that increase in luciferase means increased transcript stability of the reporter when you increase M6A. So this is a cool assay that you can use. I thought, aha, great. We can go do the same thing. So we used XHO1 and XBA1 to clone in the M6A site at 670, and this is the flanking sequence. And there's actually And this is the 670 site. There's another potential M6A site downstream of that site. And we also cloned mutant versions of each of that, either singly or double mutations in both M6A sites, both the 670 and the two sites. So what did we see? Check my time here. Okay, we're good. All right. And so this is the work of Christian Stanley from last summer, another NIDDK T35 supported student. And what he found, first of all, is that if we did a transient transfection assay into MCF7 cells, and we saw an increase in luciferase activity if we mutated the M6A site at 670. So that means less M6A, more transcript stability, which is supportive of our original hypothesis. And then likewise, if you treat the cells with metal 3, you see a decrease in luciferase activity. Treat with STM2457, see an increase in transcript stability. Mutate, you lose the effect. So therefore, less M6A, more transcript stability mediated through this M6A site in amigo-2 at 670. Yay! So, and then we did actinomycin D experiments. Actinomycin D is an inhibitor of RNA polymerase, so you can follow your transcript over time after adding actinomycin D to look at its stability, plus or minus knocking down metal 3. And by knocking down metal 3, so knocking down metal 3, we're decreasing M6A. We see more transcript stability in both cell lines, although the effect is stronger in MCF7 than it is in LCC9. And likewise, we would expect more protein, less M6A, more transcript stability, more protein. That is a direction that we can see in MCF7 cell lines, but too much scatter, it's not statistically significant. So this was 24 hours, maybe we need to try 48 hours. But we definitely see a response in LCC9 cells where we see more protein with STM2457. And then we did more experiments. We knocked down metal 3 to look at protein stability. And we see a decrease in, we see an increase in amigo-2 if we knock down metal 3 in MCF7 cells, but we don't see this effect in LCC9, which is the opposite of what we saw with STM2457. So I don't really understand that one, but we have to repeat the experiment. And then we can also overexpress metal 3. So overexpress metal 3, you get more M6A, no effect in MCF7 cells, but we would expect a decrease in stability and decreased protein, and we see the effect in LCC9 cells. So there are certainly cell-specific effects of regulation of amigo-2 by modulating M6A. So we also have validated other targets. I'm just going to flip through that because we're getting kind of close to time, because I wanted to show you what direction we're heading to now. So in summary, I've showed you that M6A readers YTHDF1 and IGF-BP2 are lower and higher in endocrine therapy resistant LCC9 versus endocrine therapy sensitive MCF7 cells. We identified differentially expressed genes and M6A differences in endocrine therapy sensitive versus resistant MCF7 breast cancer cells. We identified 74 differentially expressed genes with M6A differences in LCC9 endocrine therapy resistant versus MCF7 endocrine therapy sensitive, and 34 of those genes are associated with endocrine resistance and breast cancer progression. So we have a nice model system to keep exploring what M6A is doing. We confirmed that STM2457 reduced M6A regulated transcripts in MCF7 and LCC9 cells, and we identified amigo-2 at M6A as an M6A regulated transcript with higher expression in MCF7 than LCC9 breast cancer cells. So what's next? We hypothesized that if amigo-2 is involved in apoptosis, we have more M6A, more amigo-2 in MCF7 cells. That is allowing cells to be sensitive to endocrine therapies, whereas when we have less amigo-2 in LCC9 cells. Maybe that's involved in apoptosis resistance in response to tamoxifen, so the endocrine therapy phenotype that we see in LCC9 cells. So what can we do? We have a new undergraduate in the lab, Gabby Sanchez. She's a junior pre-med student and majoring in biology. So what she did with Kelly was to transfect LCC9 cells with an expression vector for amigo-2 that we purchased from GeneCopia. And then we wanted to examine the sensitivity of the cells to our favorite concentration, 100 nanomolar for hydroxytamoxifen, the active metabolite of tamoxifen concentrations that are found in patients taking oral tamoxifen citrate, and look for increased sensitivity and lower viability and more apoptosis in the cell. So this is hot off the press because this experiment was completed the 21st of February. So we're only a few days out. And this is an image from our Denovex cell drop cell counter right upstairs using acridine orange, which is permeable, so it binds to the nucleic acids in all cells. So that's green dots. And propidium iodide, which is membrane impermeable, and so it binds nucleic acids in dead cells, and it's a fluorescent assay. And the Denovex cell drop will count all these little images, and you don't have to try to go nuts and look at them. And so what did we see? So we're comparing cells transfected with the parental control vector versus the vector expressed in amigo-2. And I didn't put a Western blot, but yes, we've got, yeah, very exciting, isn't it? Bailey gets it. So what we can see is this has sensitized the LCC9, overexpression of amigo has sensitized the cells to 4-hydroxytamoxifen, inhibition of viability, and increasing the number of dead cells, so sending them into apoptosis. So this is a wonderful results that is good follow-up on our data. I've talked about the differential expression of genes in M6A and integration between MCF7, or well, I should say it the other way, LCC9, endocrine therapy resistant versus MCF7 cells with plus or minus STM2457 cell treatment, the metal-3 inhibitor, to identify direct versus indirect regulation by M6A. But we also have the data from the cells treated with 4-hydroxytamoxifen that we haven't explored yet. So we are exploring that, and we have a number of, so if we look at now integrating differentially expressed genes that are decorated by M6A that are differentially expressed. And so what we have, oops, this little thing is wrong in there, but we have 146 genes that are unique to LCC9 cells treated with 4-hydroxytamoxifen versus MCF7 cells treated with 4-hydroxytamoxifen. So these genes must be, or we would hypothesize that they are the ones that are contributing to endocrine therapy resistance. What are these genes? Well, here are the gene names right here. And we have picked out a few to follow up on, including Bambi, which I know that Bailey has seen before, GATA3 LDHA, that's lactate dehydrogenase, the A version that's involved in cancer, PKM, TFF1, and TXNF1. So just today I was designing M6A primers to use in the MIR glow assays. Now also, this DoD breast cancer research program grant has two parts. So I'm PI on one, and all this work is on our section of the grant. Dr. Brian Clem has the other half of the grant, and he has, and he with Mary Sumlett and members of their lab have implanted patient-derived xenografts into non-skid mice and using the MIN model. So this is from Baylor College of Medicine. This BCM 15100 is a metastatic breast tumor from a patient who was diagnosed at age 38, died at age 46 with metastasis to bone, brain, liver, and lung. The tumor was ER, PR positive and HER2 negative. This is a young patient, this is a premenopausal patient. So the cells from this this tumors, the original breast tumor, have been propagated in skit mice to develop the PDX model. What Brian and Mary and members of the lab did was to inject this into the mouse introductal, into the mouse mammary duct. And this MIND model mimics what actually occurs in human ER-positive breast cancer, which is the cells start in the duct and then fill the duct and then start to expand out of the duct to become introductal first and then invasive breast cancer. And this MIND model metastasizes to the same place as that ER-positive breast cancer metastasizes, brain, bone, liver, lung. So it's a great model. Unlike injecting, say, LCC9 or MCF7 cells into the fourth inguinal mammary gland of mice. And what Dr. Clem and Mary and the lab did was they injected the mice with either vehicle or 1 milligram of STM-2457 by IP daily for 15 days before tumor resection. And here's an example of a tumor. You can see it's in the duct and it's coming out of the duct. So, and they just, do I have the dates? Yes, you can see the dates at which the tumors were harvested. So it was a few days ago. So the tumors are now in the freezer. And our plan is to take those tumors and we're going to do single nuclear RNA-seq. We had thought we might be able to do single cell RNA-seq, but we weren't able to get enough viable cells from the tumors at the time of resection. So we've backtracked into doing single nuclear RNA-seq with 10x genomics, and we're going to do Oxford Nanopore direct RNA-seq on these tumors and look for M6A regulated genes and see if they're similar or dissimilar to those which we have detected in LCC9 endocrine therapy resistant ER-positive breast cancer cells. So I think I have tried to acknowledge the people who did the work as I've gone along in this talk and the funding sources, and I would be happy to take any questions that you have.